Good afternoon. I'm going to present you the activity of PDC Pharma, a cancer vaccine company and clinical stage having a lead asset in the non-small cell lung cancer. So the company is based on a specific technology using an off-the-shelf allogenic plasmacytoid dendritic cells, but with a scalable production process. In fact, the cell line is an immortalized cell coming from the blood of a patient having a very rare leukemia, affecting a subtype of a dendritic cell called the plasmacytoid dendritic cells that have very interesting biological property. And based on the blood of his patients, the scientific founder developed an immortalized cell line that we are using today as a vaccinal platform. So the company has been focused on the PDC lung cancer vaccine based on antigen PDC line with specific peptide. We have presented this result, the primary analysis at ESMO IO in December last year. We have opportunity to go in several other indications, including the colorectal cancer, using an off-the-shelf or personalized cancer vaccine. So we have both modality in the pipeline. We have already signed the first licensing deal in Asia with LG Chem. In terms of corporate, we are a team of 35 FTEs based in Belgium, Liège, and Grenoble, and we raised 62 million euros. The manufacturing process is clearly established and under control. So based on the cell line purified and the primary cell bank with the blood of the patients, the cell line purified from the blood of the patients, we can develop several master cell bank. And from each of the master cell bank, we can have, again, several working cell bank. In fact, it's to increase the supply to be able to answer to the market demand for ages. And when we want to launch a manufacturing campaign of the vaccine, we just have to seed the single-use bioreactor with a vial from the working cell bank. The cells grow in suspension in synthetic medium. We're using the Lonza serum-free medium, and after 9 days of culture, we have billions of cells that we split in bags, load individually with peptide, irradiate to stop the proliferations, and like for the first generation of COVID-19 vaccine, we put in a vial with cryoprotectant and ship in minus 80-degree fridge in the hospitals. So very scalable, low cost, and we are using one clone of one cell from one patient all the time. So of course we are allogenic, but we're in a kind of ideal situation in terms of raw materials. We have produced already 12 batches for the ongoing clinical trial in non-small cell lung cancer with 90% release. The pipeline is presented here. We have the lead asset PDC Lung. We have this licensing deal for PDC Lung with LG Chem. For the colorectal, with the neoantigen-based vaccine, the personalized approach, we should start the phase 2A clinical trial next year. And we have in the pipeline, in preclinical stage, over indication in urothelial hepatocellular carcinoma, ready to be launched if we get additional funding or if we have a partnership with a pharmaceutical company. The team is very experienced. We have several people coming from GSK Vaccine. We are close to the headquarters of GSK Vaccine, so it's very helpful because we are educated in terms of vaccine, even cancer vaccine, because GSK was a company strongly involved in with the MAGE cancer vaccine in the past. Next slide. Yeah. So let me show you now the clinical result of the trial in non-small cell lung cancer. The 6 antigens we have selected because it's off the shelf. Now we are using tumor-associated antigens, are MAGE-A4, MAGE-A3, MUC1, New York ESO1, Survivin, and Multimage. And for each of these antigens prior to injection to the human, we have demonstrated with the blood, the PBMC of healthy donors that we can expand effectively each of these CD8 T cells with the cell line loaded with the peptide. So the positioning, the populations, the target populations is metastatic non-small cell lung cancer with a PD-L1 in the tumor high, which represents 37% of these patients. We exclude the patient with addressable mutation And we have selected this population because the standard of care is the pembro alone. We are going to be in associations with pembrolizumab, and for the over level of PD-L1 in the tumor, medium or low, the standard of care is the combo chemotherapy and pembro. And we don't want to have chemotherapy with the vaccine because it will alter the multiplication of CD8 T cells. So we are going to compare, and we are comparing with the result of the pembro alone in the KEYNOTE-042 study published by Merck with overall response rate of 39%, median PFS 6.5 months, and median overall survival of 20 months. The design of the clinical trial, the design of the clinical trial is presented here. We have 4 cohorts of patients. The 2 first cohorts are with the vaccine alone, but it's in adjuvant setting this time, so after surgery, and in general, they have adjuvant chemotherapy. We are testing 2 doses of the vaccine, 6 patient low dose, 10 patient high dose. Here is really to observe the safety of the vaccine alone, the tolerability and the immune activity. Then we have the 2 over cohorts, low dose and high dose again, but in association with checkpoint inhibitor in metastatic patient. It is in the last cohort where we have 42 patients that we are going to compare with the result of the KEYNOTE-042. In terms of immunomonitoring, what we did is we have 4 blood sampling, first at baseline before injection of the vaccine, and after 1 week, 4 weeks, and 10 weeks post-infusion of the vaccines, because we're infusing 6 times the vaccine weekly. The clinical trial was ongoing in Europe, in France, Belgium, Netherlands, Germany, and Poland, under the leadership of Johan Vastenkis in 17 clinical centers. And let's see now the results. So we had a predefined objective for this clinical trial that was to reach a 15% increase of the overall response rate based on predefined statistical test with a power of 70% and a specific design, a Sargent 2-stage design. And as you can see here, the KEYNOTE-042 are the comparable. We reach, in terms of overall response rate, 55% for the B2 cohort, which is 16% increase. So we reach the defined objective. And for the low dose, for the 6 patients, we have reached 66.7%. So the study has been positive based on this primary endpoint. And you see here the best change in target lesions we get for each of the patients, for the 42 patients of the B2, where we have a majority of responder, and it's RECIST 1.1, so you need to have at least 2 scans, consecutive 4 weeks to validate their partial response. We have a majority of partial response, and the durability of the effect, as you can see on the right, is quite interesting. Important to mention that for 65% of the partial response patients, they have a best change in target lesion reduction over 50%, which is very encouraging in terms of prognostic for these patients. In terms of median PFS, the progression-free survival, we have observed a 36% relative increase, passing from 6.5 months for the KEYNOTE to 8.87 for our clinical trial. And for the immunodata, what I'm showing here is the result of the immunodata. We are using FACS dextramer technology, so it's really what you have in the blood. We take the blood, collect the PBMC, purify the CD8 T cells, and put in the FACS to quantify each of the CD8 T cells. So there is no stimulation, no amplification, nothing. And we have been able to observe, even at the low dose with the vaccine alone, what you see on the left, already a response of 50% of the patient above the limit of quantifications. This response is higher when you combine with the checkpoint inhibitor, is the B1. For the high dose alone, we are observing as well nice response, and close to 1% of all the CD8 T cell being PDC lung antigen specific. And where we are observing the best response are in the B2, where we have reached up to 2.3% of all the CD8 T cell being lung specific. By FACS, we can have as well the phenotype of the CD8 T cell post-vaccinations, and we have observed that all of them are effector memory, so showing that the vaccine has been definitely biologically active by transforming the CD8 T cell that are naive to memory phenotype. Another way to present the immunodata is by cohorts. Here's the fold increase. And for the A1, we have the fold increase of 3, which is the low dose alone. For the high dose alone, the A2, we have a fold increase of 20, sorry. For the B1, which is low dose with checkpoint, fold increase of 15 in average. And for the B2 plus checkpoint inhibitor, we have a fold increase of close to 35. So we see clearly a dose range effect and a synergy with checkpoint inhibitor effect. Interestingly, we had a negative control in the immunomonitoring. We have been following the CD8 T cells of the flu because everybody gets infection by flu and the level of precursor of CD8 T cell is quite high and stable. But one of the patients had a flu during the study, and this negative control became a positive control. And is what you see on the right. We have a 33-fold increase we could measure in the same clinical trial due to the viral infections. And what you see on the left, the flu-specific cell infection. And as you can observe, we have reached the same level for the cancer vaccine in terms of CD8 T cell expansion, and or sometimes even a lot higher because we have reached even 500-fold increase for some of the patients. Other important elements, we have been able to observe a correlation, statistically significant, between the CD8 T cells. We have consolidated the 3 time points, both post-vaccinations, and the PFS, which is clinical benefit, with a p-value of 0.0396, which is statistically significant. And that is all the patients where we have been able to observe immune activity. Another important element is the single agent activity. We have been able for the A1 and A2, which is for the vaccine alone, to measure the disease-free survival at 2 years because we are following the patient for 2 years. And we have seen that there is definitely a dose range effect in immuno, what you see on the left. But we observe as well, even if the number of patients are limited, 6 and 10, that the disease-free survival is improved with the high dose. We pass from 50% for the low dose to 90% with the high dose, knowing that without any treatment, it's the 2-year survival for this population is between 50 to 60%. So very interesting signal of correlation observed for the cohort A1 and A2. The safety is very mild in the B2 cohort. We didn't observe high level of above grade 3 side effect. We have just limited, just one patient stopped the clinical trial due to the treatment. Very few immune-mediated adverse events. So we can definitely conclude that the vaccine is very mild, which is very important when you are in associations with another component like the pembrolizumab. So key conclusions, so we have seen that the vaccine can provide a meaningful clinical activity and has a mild safety profile. We have a very related TAE, which is mostly grade 1 and 2, with just one related TEAE grade 4, and we have observed as well a significant response with indication of relationship with clinical outcome in the cohort B2 in association with checkpoint inhibitor. I would like to thank you for your attention, and just to tell you we are preparing a next round of funding of 55 million euros to finance the next clinical trial that will be a randomized study against pembro alone, where we will test the same regimen, 6 infusion, but another regimen which is 7 plus 6 to boost again the activity of our cancer vaccine. Thank you for your attention. It's okay? No question? Or we don't maybe have time, huh? Thank you.